Now we are going to discuss about the onion root cells observation or observation of growing root. Observation of growing root. So it means which type of tissue present in the growing parts. That's we want to know here. That's why we are doing this experiment. Okay, na? So that's why we are collecting onion root. Onion root. So in earlier before 5 days we are grown onion. We just taken one plastic beaker and we poured some water up to, and we kept a bulb of onion and to touching of the water of the beaker. So water తీసుకుని వాటర్ ని టచ్ చేయాలాగా దానిలోకి మనం ఆనియన్ తీసుకోవడం జరిగింది. తీసుకొని పెట్టడం వల్ల మనకు అబండెంట్ రూట్ ఫార్మేషన్ జరిగింది. Now we are going to show the section or going to see the structure of a root tip. Root tip. So how that cells are present in this onion. So we are going to do this experiment. So first of all, aim of our experiment is to observe growing roots in onion. Growing roots in onion. So next, objects: beaker, microscope, watch glass, slide and cover slip, etc. Okay, so that are we are using operators. Next, materials: onion root. Onion roots, that's why we grown onion. Okay, so next, chemicals: we are using water, glycerin. Water also we are using here, so glycerin. Okay, next one, procedure: a transparent plastic beaker we are taken, so is taken and filled with water. So we taken a glass of or beaker, plastic beaker, and poured some water, so filled with water. One onion bulb which is slightly larger than the mouth of the beaker is taken. So మనకు తీసుకున్నటువంటి బీకర్ యొక్క మౌత్ కంటే కాస్త పెద్దగా ఉన్నటువంటి ఆనియన్ తీసుకొని అక్కడ బీకర్ లో తీసుకున్నటువంటి వాటర్ ని టచ్ అయ్యే విధంగా మనం ఇక్కడ ఉంచడం జరిగింది. ఎందుకంటే మనకు రూట్ ఫార్మేషన్ జరగాలి కదా. సో దట్స్ వై టచ్ అయ్యే విధంగా దాని యొక్క బాటమ్ వాటర్ ని టచ్ అయ్యే విధంగా మనం ఇక్కడ ఉంచడం జరిగింది. సో దట్స్ ఏ. నెక్స్ట్ వన్, it is kept on the mouth of the beaker. Mouth of beaker, the growth of roots are observed for the few days. Few days మనం అలా ఉంచినప్పుడు మనము growth ని అబ్జర్వ్ చేయడం కూడా జరిగింది. నెక్స్ట్, one of the roots tips is taken from an onion bulb, so it is placed on slide and glycerin is put in extracted root tip. The part is covered with a cover slip, so the entire mounted part is kept under the microscope and observed. Okay, so that's it. First of all, we want to do this experiment. Now we can separate the root tip from the bulb. Okay, we will take some water in a watch glass or petri dish. So we will separate root tip of the onion bulb. Okay, so you can separate it. So now we separated root tips of the onion bulb. Okay, so we collected in a petri dish. So then we can take a clean slide, so we can keep this root tip on the surface of the slide. Okay, we kept on a slide. So now we can pour a small water drop. So and then we will take glycerin. We can take glycerin in it. So we can pour a drop of glycerin on the surface of root tip what we collected here. Okay, so then we can place a cover slip on it. So this is the most important one. We can place a cover slip. Okay, so we can place a cover slip. Okay, yes, so we can use a brush to rub the or to spread the onion tip in on the slide. ఓకే, ఆ స్లైడ్ పైన రూట్ టిప్ అనేది కాస్త స్ప్రెడ్ అయినట్లయితే మనకు విజిబిలిటీ అనేది టిష్యూ అనేది క్లియర్ గా కనిపిస్తుంది. ఓకేనా, దట్స్ వై వీ కెన్ యూజ్ అనదర్ ఎండ్ బ్లెండెడ్ ఎండ్ ఆఫ్ ది this brush and we can gently press the cover slip. Okay, so now this is a slide we prepared, so we can observe this one under microscope. Okay, so are you understand? Yes, so this is how we can make the slide. Okay, so now this slide we can observe under the microscope. If you observe under microscope, we can see the structure of onion root tip likewise. Okay, it can show a root cap, and above the root cap we can see the region which are having thin membranous tissue. That tissue is known as a meristematic tissue. So these region are dividing region, cell dividing region we can observe there. So that cell dividing region have a tissue that is a meristematic tissue. That meristematic tissue is responsible for cell division or growth of the root. Okay, so it means different parts of the plant body can show the different kinds of tissues. We observed leaf peel and we observed onion bulb, and then now we are observing the structure of the root tip. Okay, so it means the plant body, different types of parts of the plant body can possess different kinds of tissues. Okay, mainly the plant tissues are mainly 4 types. Okay, so mainly first one dermal tissue, next one meristematic tissue, third one ground tissue, fourth one vascular tissue. Don't forget. So first one dermal tissue, meristematic tissue, ground tissue, vascular tissue. Okay, now do you understand up to here? So then we will discuss about the different kinds of tissue of plant body. So this is the root. So this is the root tip observation or root observation in onion bulb. Are you understood? Yes, sir.